give the floor to Vineeta, just one announcement. So those of you who are planning to submit your travel reimbursement forms, could you please fill them up and have them ready tomorrow so that around 11 o'clock, someone from the office is going to come here and you can give the travel reimbursement form to him. Okay, so without further ado, let's invite Vineeta to continue on her talk on the immune system.
Okay, so a couple of things and maybe questions, since some people wanted to ask questions and we broke for lunch. So one explanation, information I should say. We talk about incidence of a particular disease. It could be infectious disease, non-infectious disease. We also talk about births and deaths and stuff. There is a finiteness in the birth and the death. I mean, a child is born and the birth is recorded. A person dies and the death is recorded. Infection doesn't fall into that category. So when people talk about incidence of tuberculosis or incidence of dengue or malaria or something or the other, there is this gray zone of whether the infection is being scored or the disease is being scored. And as I was explaining to somebody after my talk, that the zone between infection and disease is gray. If I feel I am really unwell and I go to see somebody, that becomes the disease. If I have phenomenal tolerance to minor pains and this and that and I don't go, I will never be recorded as a patient and hence a case or a disease. So I will not get counted as contributing to the incidence of the disease. So this is something that has an arbitrariness and it needs to be borne in mind, and all I was pointing out to is that when you get data for incidence of here and there per X many population, try and figure out what is the methodology that is being used. So if it is based on, say, presence of antibodies in whole population or a subset of population like national sample survey kind of population, then you are actually looking at infection plus disease together. If it is simply by hospital admissions or private practice or admissions or treatment, then it's, or in all probability, it is the actual disease which is getting counted. And these two should not be equated, especially if you want to use these solid figures for projecting whatever you would like to do for building the model. So on that score, I'll move on to the sort of next part, which is here. So, I mean, I know the divisions between what parasite does and what host does are not really watertight when I'm talking, but at least there is some distinction that I was making. So now I'm looking at, sorry.
Different phagocyte, neutrophil, macrophage, lymphocyte, etc. So all of these together are WBCs, and they're separated into two parts. One is neutrophil macrophage, which is part of the innate immune system, and the other is lymphocyte, which is TNB, which is part of the adaptive immune system. Is that correct?
Yeah.
Okay, and macrophages are, neutrophil is the first line of defense, followed by macrophages, and macrophages cut up the invading bacterium, let's say, and display little parts of it on their surfaces to be recognized by the TNB.
Yeah.
That correct? Did I get that right?
It is correct. Okay. So the connection I did not make so explicitly, but I was tentatively making that when these cells phagocytose particulate matter, as in bacteria and stuff, viruses, these entities, now I will talk about that, what happens once there is a phagocytosis. and how that contributes to T and B cells coming into picture, and hence the connection. So, these are not watertight compartments. That is why I said that if you do not have innate components, you do not get wonderful response with T and B cells, and that I referred to, did not emphasize. You will get to hear it in this part now. So, anybody else who had a question which wasn't answered? Don't have to, just thought before you completely forget what it was. All right, so we will move. So, this is the scenario post invasion. So, now we are looking at the parasites which are inside the host body, and now we want to understand how the immune system would locate them and mount appropriate immune response. Most of this is focused on T cells, but I will add up bits and pieces here and there about B cell responses as well. So, all you have to remember is that when I say B cells respond, B cells would look at the contour or the shape of the protein which is not part of itself meaning the host, and would mount a response. So, B cells would divide, and this division is normally unequal division, in the sense morphologically also one can make a distinction between memory B cells, so that those will persist for a longer period, and a cell which is called a plasma cell. The plasma cell loses its ability to divide extensively, while memory cell retains its ability to divide, so that it will come into picture next time the same organism comes to the host. The plasma cells sit at different locations, and either they are capable of secreting antibodies which are for a few weeks or for few months to few years. So, we do not know enough about these short-lived and long-lived plasma cells, but that was the reason why I was mentioning earlier that if I had an antibody generated against, say, smallpox, because I remember getting the not the disease but the vaccination against smallpox. So, while those responses are mainly T cells, when I was young, I am sure my immune system must have mounted responses against smallpox, not cowpox virus. Will the antibodies be detectable today? That will be a question. This is a known invasion that was introduced, but whether my body will have the antibodies to the cowpox virus today, and I suspect it may not, or if it has, it will be in really small proportion, and that would depend primarily on the plasma cells which were generated, because there is no exposure to smallpox. Smallpox has been eradicated, at least since 1975 there has been no exposure to smallpox to any of us. So, from 1975 to 2016, will antibodies be detectable or not? These kinds of questions are important, and this distinction between memory B cell and an effector cell or the antibody producing cell generated by B cells is very distinct, and that is not what you see in the T cell compartment. Those are some of the other differences also exist, but let us look at the parasites and the locations and what happens. So, this is a cartoon, is really a cartoon, and you will appreciate that neither Satyajit nor I have real skills in drawing. However, it is useful enough to use it for teaching. So, take this red ring as a cross section of a blood vessel. Okay. So, it will have white blood cells, it will have red blood cells, and what you see these creatures as are the invaders which are currently at that time point in the blood vessel. So, area outside is what is described as tissues, and these tissues can consist of variety of cells. These could be in the spleen, it could be in the liver, it could be in some other locations, it could be brain, anywhere. So if there is a set of bugs which are inside the vessel, you have to remember that I can't quote the actual flow rates, but from the artery, when the blood is moving towards the periphery, it is at a relatively rapid rate. So the center of the blood vessel will have very high velocity stream of components flowing towards the walls. There will be somewhat less velocity that you will see. So these bugs can get pushed out of the blood vessel, especially when they go to the small capillary vessel kind of area, and then they can be outside the blood vessel. So what I am trying to highlight is that even when from this early stage they get out of the blood vessel, they can continue to sit outside the cells, or they can sit inside the cells in at least two different ways. And what is shown here is that there is a vesicle inside a cell, and what is shown here in this cell also there will be a vesicles inside the cell, but this particular bug has chosen not to go into the vesicles but sitting inside the cytoplasm. I know I haven't shown nucleus anywhere, the cells do have it. That's why I said that this is not necessarily the great picture, but there will be nucleus and there will be cytoplasm, and these are the vesicular structures. Here also there will be a nucleus, vesicular structures. Some of the organisms will sit not in the vesicular structures but in the cytoplasm. So relatively ill-defined area, non-compartmentalized area. So the way immune system handles these specific locations and the organisms sitting in there is sort of, I shouldn't say predetermined, but it makes sense when you think of the logic. And that logic is something that's explained here to an extent. And so these are the strategies. So now, sorry, this works, yes. So now we are looking at my voice has to be modulated. Anyway, so my voice is working, yeah. So these are the extracellular parasites, extracellular invaders, and these are the cells which are encountering them, meaning coming in contact with but not necessarily engulfing, not necessarily phagocytosing them. So what facilitates phagocytosis? So one thing is there is an efficiency. So if there is simply a particulate matter, it will also get phagocytosed. If it is coated with one of the two things or both the things, as I will explain in a second, the efficiency of phagocytosis goes up. So it's not that neutral particles are not phagocytosed, but bacteria which are either coated with complement, that's one of the soluble factors which is found in our blood all the time, or bacteria which are coated with antibody are picked up by phagocytes much more rapidly. So we are talking about rapid clearance. And you can imagine that complement will be present throughout. Antibody directed against that bug will not be present when it is first exposure, right? Body is seeing that bug for the first time, so there is nothing like pre-existing antibody. I know I'm making generalizations, but this is only for Jose, who should not object. So this is the antibody is antibody mediated enhancement of uptake is in after the primary or post primary immune response scenario. Enhancement of bug because of complement mediated uptake is present during primary infection as well as in secondary infection, because complement is part of the innate immune system. It is always there, and as I said, red flags are identified of sorts. So complement will be contributing to that, and complement is one protein family of proteins, multiple components, and they come together and actually bring complement mediated lysis. So it's hard to explain in 2 minutes, but essentially if the complement bind, many cells secrete complement. Bacteria, our own cells secrete complement. It's not because of the presence of bacteria. So we have complement in an inactive state, multiple components of the complement in inactive state in our blood all the time, and when there is some trigger, then the complement gets activated. When it is bound to the bacteria or something, then bacteria or some other particles, then there will be complement mediated rapid uptake by the phagocytic cells. So how complement components are kept in a non-active state is a very complex field of research, and I don't work on it, and it's really too complex even for me to give you a quick answer. Between complement and antibody, the extracellular organisms, microorganisms, are efficiently picked up by the phagocytic cells. So that clearance is achieved. So we talked about the extracellular organisms. Now we are talking about two kinds of intracellular organisms. This organism, which is sitting inside the vacuole, you can imagine that if I can get rid of the vacuole, I can get rid of the bug, right? However, here there is no way to get rid of the vacuole, or nothing that separates the bug from the rest of the cell. There is no lining as it is here. This is another membrane also. So the strategy that gets used is that this bug sitting inside the vacuole in the phagocytic cell will be killed by means so that there is outpouring of some of the enzymes, reactive oxygen nitrogen species, and various enzymes are concentrated here, and because of which intracellularly, intravesicularly located bacterium is killed off. So this is a strategy which is used by, which is helped by certain kinds of cells. These are called the so-called helper T cells, CD4 T cells, CD4 helper T cells, and so on and so forth. I know we use too many loose nomenclatures, but this is one of those, one of the many. So helper cell signals will help in intracellular parasites inside the bubbles, as it is called, vacuolar parasites. These are non-vacuolar parasites. So killing them is a different way, task altogether. Here, if you kill the cell, the bug will automatically get killed. If you do not kill the cell, the bug can sit inside, replicate, and maybe the bug will kill the cell rather than the host immune system killing the bug. So if one is looking at conservation of the host energy, because you need not just ATP kind of energy, but yes, that primarily, energy to survive and combat infection, this is keeping the cell alive but keeping the bug, killing the bug. This is keeping the, not keeping the cell or the bug alive. And these are called as killer cells, and the T cells of this category all are called cytotoxic T cells, CTLs, or CD8 T cells. So these are the kinds of strategies which are normally used, And mind you, this is not every bug that comes that enters the host is capable of eliciting all three, maybe a little more of this, little less of that, and so on and so forth. So for example, if I look at influenza virus response, influenza virus specific response, there will be so-called CD8 cytotoxic cells, there will be CD4 helper T cells, and there will be antibodies against influenza virus that will be found in the same individual. Some individuals might have more antibody, less CTLs, and more TH helper cells, and a combination thereof. So it's not exclusive responses, but what is seen as a logic is that if one can save the cell but still kill the parasite, this approach may be really useful for those parasites which are sitting inside the vacuole. If one cannot do it in this fashion, but one still has to kill the parasite which is sitting inside the cell, then kill the cell off. At least further spread can be prevented. So that is why one thinks of this or that or these two kinds of approaches. Got the answer? So as I said, these are broad generalizations, but these are the three categories of responses that one tends to see. Let's go through the details of the previous one to highlight which kinds of organisms are commonly seen in these locations. So let's look at these extracellular, staying outside the cells. These invaders are the kind which cause pneumonia. So I am talking about Bacterium streptococcus pneumoniae. Meningitis is again meningococcus. Sore throat is, I can call it streptococcus. There is also staphylococcus. We are also talking of malarial and filarial parasites which can be sitting in extracellular locations. So these are a variety of organisms, and while I am happily telling you these are the ones, yes, this week, sometime earlier this week, no, today is Monday, sometime earlier last week, I read a paper where it said that staphylococcus sits very steadfastly, especially the MRSA kind, multi-resistant, drug-resistant kind staphylococcus sits inside the cells, and that is why it is resistant to antibiotics that are given, because antibiotics do not necessarily reach inside, and so on and so forth. So consequences thereof. So while one likes to make generalizations even about invaders, with time one has to revise one's knowledge and say that, oh, maybe this also happens, and so you obviously need to think of different contributions of the immune responses. So
is there like some generalization thinking about what microorganisms or what pathogenic microorganisms can survive outside the cell?
yeah, so in a minute it will become apparent. So I said here these were mostly the bacteria that I talked about, except for malaria, which is unicellular, and filarial parasite has a very complex life cycle. So there is microfilaria which is found in the blood. It is much bigger, micro level, and there are also adult worms which produce these microfilaria. So the parasite, as in malaria parasite, filaria parasite kind of cycles are much more complicated as compared to any of these. So many extracellular bacteria, as they would be called, some of the fungi would fall into this category, that they may remain extracellular, and antibody mediated and complement mediated immune clearance will be the most beneficial immune response to be mounted by the host.
So how do they get nutrition? Like if you don't sit inside a cell?
But I mean, there is body fluid all over which will have protein.
So they just catch it.
Yeah, and they have capacity to use their own internal enzymes. So if you are looking for amino acids, if you are looking for sugars, carbohydrate sources and so on, you will find them in the body fluids that in which they will be found. So this is looking like this is in vacuo, but it is not. It is inside the cell, sorry, intercellular spaces, if I can call that, not inside the cells. So there is a second category which is this, and there is a third category which is this. Maybe I'll go to the next.
How is the antigen and the host cell determined? Like, is it through evolution? There are certain viruses or bacteria that infect particular cells. How is this actually being determined? Is it through course of evolution, those receptors that are there on the antigen and the cells?
There is no deterministic approach that one can talk about in the immune system. All one can do is how does one explain this? So that's why I said the logic that I am trying to put forward here, and that your question maybe I'll address outside the classes, because that will, I was not thinking of covering that, but if I do, I'll also address that. But when we see, for example, certain kind of immune response is protective in tuberculosis, or protective in leprosy, or protective in flu, how do we rationalize that observation? So that's how one thinks that, yeah, it makes sense. So it makes sense is what I'm trying to tell you. But many times what we see in reality does not make sense. So if we had this, it would have made sense, and I'll show you in the next 3 slides what I mean. All these 3 things that I'm expanding, that's why I thought that this was the extracellular organisms about which I started talking, and I know this is catch hold of them and destroy fast. So that is where the complement and the antibodies would come into picture, because eventually with nutrition available even in the tissue fluid, these organisms will multiply. And once they multiply, there will be what is called as sepsis or toxic shock, and these kinds of severe illnesses, some of which are associated with, say, streptococci or staphylococci. So those really can be quite deadly. So in children who get infected with either pneumococcus pneumonia or Klebsiella pneumonia or even meningitis, quite a few of them really succumb to infection or are left with some of the consequences which are not life threatening, but it is morbidity, it is sort of being handicapped. So malaria and filaria also, as I said, for different reasons fall into the category. Filarial parasites are so big that they simply cannot get into any cell at all, because they are bigger in size than the normal cell. And I should probably mention this, that neutrophils, lymphocytes, mostly actually lymphocytes which are at resting stage are about 7 micron in size, 7-8 micron in size. Neutrophils can be a little bigger, and monocyte macrophages, and I did not introduce the word dendritic cells, but even that category is there. Those could be sort of 10 to 15 microns in diameter, in its diameter. So if you have a 20 micron or 30 micron microfilaria, as in filarial parasites microfilaria, it cannot be engulfed. That is something that one has to realize, that the phagocytic cells also have a limitation, that they can only engulf things which are smaller than them. So this I had already mentioned, that these kinds of extracellular organisms are phagocytosed by neutrophils and macrophages, and they are also complement and antibodies which help in the clearance by the phagocytic cells. So the second category which I had mentioned was those sitting inside the bubbles, and these what we describe in terms of microorganisms as facultative intracellular parasites, FIPs. These organisms are perfectly capable of using their own enzymes for producing whatever they need, but they will obviously use the amino acids and sugars and whatever else that is required as the basic ingredients from the host. So inside the vacuole, whatever nutrients are available, those will be used. And the usual way the phagocytosis operates is that the bacterium or the particle is engulfed, so there is a vacuole formed now, vacuolar material, and this will have approximately neutral pH, because outside is this pH about 7 and from outside it's engulfed. So the vacuole as it gets formed has approximately neutral pH, but slowly and steadily it by certain mechanisms its pH drops and it moves and fuses with what is called as the lysosome, and lysosomes have pH of about 5.5. So phagosome lysosome fusion is a deadly signal, in the sense lysosomes are full of enzymes which will act on and degrade the organisms. There are also possibilities of having INOS, reactive oxygen and nitrogen species, so that as soon as the phagolysosomal fusion happens, the chances are that what is sitting inside that fused vacuole will die. So one strategy that some of these organisms actually use is that secrete certain products as soon as you get into the phagosome, so that phagolysosomal fusion is prevented or at least delayed. During that time, one can actually find that these bacteria can divide inside the cell, inside the vacuole. So that becomes a niche, and now you can imagine that even if you have lots of antibodies and complements which are floating outside the cell wall, cell membrane, they are completely useless, because inside the membrane, inside the cell, these bacteria are happily sitting and multiplying. So these are called facultative intracellular parasites, and the ones that fall into this category are Salmonella typhi causing typhoid, Mycobacterium tuberculosis, Mycobacterium leprae, and Kala Azar, which is the leishmaniasis that we are talking about. So what is it that happens? As I said, there is support. Once the phagolysosomal fusion is induced, then there is support for energy which will help in killing of these bacteria. And I'll give you an example. There is M. tuberculosis, which we will probably talk about. So M. tuberculosis falls into this category, and virulent M. tuberculosis, which is known to cause disease in humans, is actually one of the great evaders of phagolysosomal fusion. If you take another cousin of Mycobacterium called Mycobacterium W, it is a parasite, what do you call it? Non-pathogenic microorganism. If you have macrophage, this is now model systems, in the sense experimental model system, not mathematical. So you have macrophages, you add M. tuberculosis, you have macrophages, you add M. Mycobacterium W, they both will be phagocytosed. Few hours later, you try and remove all the extracellular bacteria and see what is surviving inside the macrophages. You will find M. tuberculosis positive, MW negative, which means while both were phagocytosed, MW got killed off and MTB survived. This strategy of surviving inside the vacuole actually is considered as a virulence factor, in the sense that it helps in being tuberculosis being virulent, surviving inside the cell, surviving inside the host, and obviously later on spreading from one cell to another cell after infection. So let's look at the third category, and this category is normally the one where the separation between the vacuole is not imminent. And so the organisms are normally sitting inside the cytoplasm, and many organisms which do not have self-sufficiency in terms of its own enzymes tend to fall into this category, and very, very commonly found category is viruses. As you know, viruses are called obligate intracellular parasites. So, why are they called obligate? Because unless there is a host and a host machinery to synthesize their own proteins, nucleoproteins and whatever else is required, they cannot replicate. If you leave the virus outside, it's as if it's a dead, non-viable, non-living particle. It can't do anything on its own unless it gets into the cell. So, these are called obligate intracellular parasites, and obligate intracellular parasites normally use this strategy, that they will sit in the cytoplasm, then there will be endoplasmic reticular and other machineries which are available for protein synthesis. Their own proteins will be synthesized very often at the cost of host protein synthesis. So, you may see an infected cell, virus infected cell with many virus particles inside, which will bud out one after the other as one way of replication. So, for all of that, the virus, the host machinery is absolutely essential, and because there is no membrane associated separation that one can see, there is normally killing the cell as the least damaging or most benefiting strategy to use. This is where I was mentioning earlier that if you have viruses, then using cytotoxicity as the most efficient killing system makes sense, because the viruses need to be gotten rid of and you can't get rid of them in any other way. If you have facultative intracellular parasites, then why kill the cells? Get rid of the bubbles, get rid of the organisms, but that same cell might be useful. Macrophage may be useful to kill another parasite and another parasites, because macrophages, neutrophils die very fast after their production. Macrophages can survive for weeks and months. So, it's useful to have those cells surviving rather than creating another cell and another cell. It's essentially conservation of energy. So, and if you do not have intracellular but extracellular location, it makes sense to have all those floating antibodies which can tie up with the particulate matter and it gets killed off. So, all of this in retrospect, when we know this, these are the most protective responses that one has seen, it makes sense to explain it like this. How has it developed? I have no answer, and I don't believe in God. So, if you can say God has done it, I can't do that. I can't say that yes, that's how it is, but describing logic and rationality in this context is what I am trying to do. So, these are the three categories of organisms that we look at and decide that this can be used. But as I said, and I gave influenza example there, that in influenza, because it's a virus and it will bud and move from one infected cell to another infected cell to stop spread of infection, it's best if one can kill the infected cell. However, the particle which is released may go and actually encounter the antibody which is already generated. So, it's not the case that one will not find other responses mounted by the body, but if I have to choose one kind of immune response that I would really like for protection from influenza, I would still say cytotoxicity, because even if there is neutralizing antibody, it is useful in the initial stages. But what if I do not have neutralizing antibody at all, or is in very small amount and the virus load is too much? It's all gone. It's gone beyond the neutralizing antibodies neutralizing capacity. Then what do you need? Antibodies are no great help then, and then you need cytotoxicity. So, this is one way of rationalizing what kind of immune responses one would like, and what is ultimately there, and this is where the models are useful, but they are completely inadequate, and I am talking about experimental models. I don't work on mathematical models, though I participate in these conversations. So, if one is looking at the animal models, changing one parameter at a time to see what is protective response is a possibility. so that I am eliminating other variables which are found in human beings. For example, somebody may have had earlier exposure, somebody maybe a woman might be pregnant and her immune system may be currently compromised, somebody might be suffering from cancer and hence partly immunosuppressive, somebody might be too old. We are all genetically diverse. So if I want to look at immune responses to influenza in such heterogeneous population, arriving at inferences is harder. What one sees is the associations. Instead of that, if one uses experimental animal models, then it's possible that I am keeping all the genetic heterogeneity to 0 and looking at infection outcome. Then at least I know, is there a genetic component, is there not a genetic component, which are the other components which are important. So that's why when I started talking in the morning, I had made multiple ways, multiple choices about the modeling in infectious diseases. So while I did not mention animal models there, but looking at using animal models also has its great advantages and limitations. So, okay, now I'm going to stop just now and ask people to ask questions if they have, and maybe 5-minute break, and then one can delve onto mycobacterial infections.
Viruses mostly within cells, not within vacuoles. Bacteria within vacuoles, and also some of them in the intracellular medium, and parasites essentially intercellular medium. Is that like a good generalization, or are these their niche that they occupy within the body?
In some cases the generalization is correct. If viruses, as I said, because of their obligate nature, they have to have cell as the anchor. So even if you find viruses which are circulating, it's not that you don't find them, but they are not the ones which are actively replicating. So they are getting transported from here to there to there to there. So for viruses, their presence intracellularly is mandatory for long-term survival of the parasites, as in viruses. But for the bacteria, and I did not include fungi here, but I also included Leishmania, which is a unicellular parasite, or there are amoebae and other parasites as well. So many of these can be falling into different categories, and reason why they fall into this versus that is hard to predict. But if they are going to reside extracellularly more, or found extracellularly more often than not, then it makes sense that antibody mediated clearance will be useful, because they are unlikely to go inside the cell and find that as their permanent abode. If you are looking at Leishmania, Salmonella, Mycobacteria, any of these as facultative intracellular parasites, then because they need to be, they multiply much better inside the cell, then their outside existence is also incidental. And that's why having a response which will kill their intracellular location and that as their niche will be the most beneficial, as I would see it.
Okay, but what about within vacuole versus just inside the cell? Advantages versus disadvantages?
Viruses do not, well, viruses are small in size as compared to facultative intracellular parasites. They tend to attach to some of the cell surface receptors and or simply invade the membrane, depending on different types of viruses. So it is also seen that even if they are initially seen inside the vacuole, they have a tendency to go out from the vacuole to the cytoplasm, because the synthesis machinery is better available, I suppose, in the cytoplasm instead of the restricted vacuolar space, because there is no RER endoplasmic reticulum kind of access or available in the phagosome lysosomal vacuoles. That is why if they need it, they have to be in the cytoplasm ultimately. So that's the way one would rationalize it. It's not, that's how it has been evolved. I don't know that.
You mentioned the pH difference between the inside of the vacuole where the bacterium is exposed to versus in the cytoplasm. How big is this? What is the
7 versus 5.5. And what you have to remember is that different enzymes will have different pH optima. So enzymes which are optimum at pH 7 will obviously, not obviously, but are likely to be very minimally active or suboptimal at pH 5.5. So what is shown is that lysosome has really major package of enzymes, proteolytic enzymes, which are reactive and bring about a lot of degradation of all kinds. Here I am talking about facultative intracellular parasite, and I did not address the earlier question of antigens and processing and so on. But these phagolysosomal vacuoles or endosome lysosomal vacuoles are the major sites of proteolytic degradation for all of these organisms, and the peptides which are generated in these vacuolar compartments are the ones which get loaded onto what I hope you know the terminology, HLA or MHC molecules in those locations. So that is the function that also is evolved. And what I did not highlight, and maybe I can go back and say, is that when I was talking about CD4 T cell function, for example, maybe I'll go back to that slide. Okay. So when I was talking about CD4 T cell function here or CD8 T cell function, what you have to realize is that there is no trace of existence of this organism outside at the beginning of the infection. It's just phagocytosed. There is nothing which is outside to tell that this is an infected cell and adjoining cell is an uninfected cell. So what CD4 as well as CD8 T cells recognize from outside is the presence of a bug inside. So for that, what is required and what is not really clearly shown here is that whether here or in this location, the enzymatic machinery of the cell, which will have different optima, 7-ish here and sort of 5 and 6 here, they will generate different sets of peptides because the enzymes working are different. And the peptides which are generated will get loaded on HLA molecules or MHC molecules as we know them, and they will come outside. So this particular cell will have thousands and thousands of MHC molecules in any case as a homeostatically existent property. If this cell is infected, a small subset will have peptides which are coming from this entity. Similarly in this case also. So that will make a distinction between an infected cell and an uninfected cell. So that's how this particular cell, as it happens, this is a yellow cell which is recognizing this. But if a blue or a red or some other cell is actually looking at this, it will not get the signal. Say that this is an infected cell, because this particular T cell's receptor is incapable of recognizing that peptide MHC complex. So these both CD4 and CD8 cells tend to recognize presence of intracellular pathogen by their remnants which are associated with MHC molecule on the surface of it. So that makes the distinction, and that is why the complicated process of proteolytic machinery, having the synthesis of in the endoplasmic reticulum of MHC molecules, these peptides being there for loading and going out on the surface is sort of all linked with this kind of detection system, which I could have explained, but I did not at that point. Yeah? Sorry? B cells. No. CTLs will actually just recognize in this fashion. So, you are saying working of B cells and CD8 T cells.
CTLs.
CTLs is CD8 T cells, cytotoxic cells. So, this is a cytotoxic interaction that I have shown, and this will result in this cell killing this cell. That is the cytotoxicity that we are talking about. If you look at the B cells will recognize, for example, I have shown all of these as very smooth surfaces, but if you look if you increase in size, all these microorganisms will have certain shapes on the surface. Those shapes can be directly recognized by a receptor on the B cell. Okay. So, B cell will recognize those, and for that they do not need the cell association. Right. This T cell receptor requires cell to cell contact. The B cell does not require cell to cell contact. It will recognize even a free floating objective, and if it is appropriate, it will start responding. Then the cells would divide, differentiate, and antibodies will be generated.
So, could we say that that B cells work for virus, means here this is for infected cells. So, B cells work for viruses, is this true?
B cells can work for anything. For example, if you give something that is not even a danger normally, say ovalbumin protein, egg albumin protein, okay, it is not a danger, I mean it is not going to kill the body, but it is a foreign entity. So, in appropriate form, when it is introduced into the body, it will also be recognized. So, B cells would look at ovalbumin protein, some of its shapes will be recognized, and there will be an anti-ovalbumin antibody response. Similarly, if there is a killed virus, for example, the virus which was here, the cell got killed, and in some situations the virus particles which were already there got released, or viral proteins got released. Even those can be recognized by B cells and response mounted. So, B cells will mount response by preparing antibodies or generating antibodies against anything which is sort of floating around, provided the B cell receptor is capable of recognizing it. So, 1 in a 10 million argument that I was making earlier, that still holds true. So, that particular B cell to be present near the appropriate antigen or appropriate protein or bacterium or virus is going to take time. I mean, it's a moving population, so that you do need to come in contact. If there is no contact at all for the 5 days, for example, there may not be an antibody generated against that particular antigen, but there might be antibodies generated against 5 other antigens which are forming part of the same virus.
Thank you.
Okay. Move on. Okay. So, all right. So, this is sort of a major bugaboo, and I like to talk about it because it is a completely unresolved issue. Immunologically, it is not possibly so easy to do modeling on mycobacterial infections in India as against mycobacterial infections in developed world, because the conditions are different. That is the reason why I thought I would probably bring it up and simply throw it open for discussion. And keep in mind, I have been talking about infections and modeling and everything. Currently, the only minority work I do on infectious diseases is about Japanese encephalitis virus. It has been many, many years since I have done any serious work on any infectious disease. So, it might look like, why am I standing on my hind legs and talking about it, but what the hell. So, it is some information which is available, and once upon a time my specialization was in microbiology, and now so many years in immunology. So, infectious disease still happens to be an area of my interest, though I do not necessarily know in depth about many infectious diseases. So tuberculosis, and mind you, when I say tuberculosis, I also mean at some point I'll come back to leprosy as well, hopefully. Don't remember now as slides because I was supposed to talk only for 2 hours. So it has become extended now to 5. So I just put some slides together. So now it is going to be more disjointed. So tuberculosis is caused primarily by M. tuberculosis. But even in India, the M. bovis, which is a bovine mycobacterial species, can cause tuberculosis. Once upon a time, I don't know how many years ago, but before milk got pasteurized, if there was cattle which was infected with M. bovis, then in the milk there will be mycobacteria M. bovis, and those, if we drink, there was a possibility of higher incidence of catching M. bovis mediated tuberculosis in all sorts of populations. So, however, once the pasteurization of milk has been introduced, and most of us who live in urban areas never see cows giving milk or buffaloes giving milk, but we only see the containers. So, all that milk is pasteurized, and hence M. bovis causing tuberculosis as a major cause of tuberculosis has gone down. However, H. Krishna Prasad in All India Institute of Medical Sciences has been working on many years on tuberculosis, and he has actually documented that in human population in India, obviously, there is not just M. tuberculosis infection, there is a double MTB, M. bovis infection, as well as M. bovis alone infection. So it's not something that we can ignore. However, the symptomatology is not really different. So KP's diagnostic measures were PCR and other things, because cultures are not particularly different and so on and so forth. So it's a PCR based diagnosis that had helped him to arrive at this milk. We don't know. Earlier it was milk. Now is it secretions? Secretions is one possibility. The other is people are working with cattle, as in farmers who are people who are breeding cattle. If those are infected, then there is a possibility. But that's why I said it's not a non-existent disease, but clinically it's not necessary to diagnose it as separate unless you want to treat something separately. But cattle tuberculosis is quite common. Monkeys can easily get infected with tuberculosis. Sorry? Yes. Yes. And in fact, BCG vaccine that we talk about is M. bovis BCG. So, it was a cowpox, again, it's coming to cows and cattle. Smallpox vaccine that we talk about was a cowpox derived virus. Here also, the BCG vaccine is a cow derived bacterium which is used, further developed as a vaccine. Okay, so tuberculosis is a complex disease for a bacterium. And when I say that, I have filariasis as an example, or Kala-Azar as another example. There, the complexity is a lot more in the parasite, because there is this form of the parasite, that form of the parasite. In malaria, there is a liver stage, in the blood stage, and so on and so forth, and it goes out into the vector, comes back again. So the life cycle of parasites is much more complicated in malaria, in leishmaniasis, filariasis. In tuberculosis, it is the host spectrum which is quite amazing and confusing. So there is something called primary complex, which earlier, again, I realize how old I am, because BCG vaccine was not compulsory when I was young. I probably got it for the first time when I was 25 or something. But it was in those days when BCG vaccination at birth was not compulsory in India. There were many children who came down with what is described as a primary complex. So between 0 to 5 years, mostly actually 0 to 2 years, young children came down with tuberculosis. That was called primary complex. In many instances, with treatment, it was cured and it disappeared, in the sense that these kids never suffered from tuberculosis again. But many of them also did suffer. Now that we have BCG vaccination, and as Sebastian had shown earlier a graph of measles in UK, that there were these waves of episodes, whatever cycles that were there, every 2 years there was an epidemic, and as soon as the vaccination was started, it is more or less flat all over the place. That is the case about primary complex. It was not periodic and episodic, but it was pretty common before BCG vaccination was introduced. So now if you look at or ask any of the medically trained people who are coming out of their degrees now, how many of them have seen primary complexes? Probably none would see it. But people like me had seen it, despite never practicing seriously as a doctor. But during my training, I had seen it. So the frequency was that high, is what I meant to say. That primary complex was there, and then there was a post-primary pulmonary tuberculosis. This post-primary pulmonary tuberculosis, unfortunately, is still probably occurring at the same frequency today, post-BCG introduction, as it was 50 years ago or maybe 100 years ago. I don't know whether we really have data or not. And mind you, while I'm talking about tuberculosis, the actual numbers I will try and not quote. I will only talk about trends, because I haven't looked up actual numbers. So rather than making mistakes of telling you 5% and it's being 25, I will not quote. So even if I by mistake I quote something, take it with a pinch of salt, because I haven't looked at any recent data about tuberculosis from this perspective in terms of quoting the numbers. And I know that when I talk to modelers, infectious disease modelers, the numbers is the primary consideration for them. I am a biologist, so I can just hand wave and say these are the trends. So I'll do that happily. So primary complex and post-primary adult pulmonary tuberculosis are, as I said, one is pretty much disappeared post-vaccination, the other remains as it was earlier. And there are also extrapulmonary forms of tuberculosis called lymphadenitis. So the lymph nodes that I had shown you in the cartoons, those get inflamed. So there can be these lymph nodes here or some other lymph nodes. Cervical lymph nodes are very, very common, and those are also all swollen up. And there is tuberculous meningitis. There is genital tuberculosis, as in uterus is infected with tuberculosis, and in many cases such women become infertile if it happens in the early reproductive age. There is bone tuberculosis, and some people might have seen what is described as pot spine. That also has gone down significantly, but if there is a bone which is infected, especially the bone of the spine, then there is a fracture which takes place, and because of which there is a permanent disability which can occur. That's also bone tuberculosis. There is intestinal tuberculosis, and one of the serious forms is miliary tuberculosis. And the reason miliary tuberculosis is at the bottom actually is that post BCG again, miliary has gone down significantly in India. So what is the mode of exposure? I did not talk about respiratory route of exposure. I showed you images of the gut, and I showed you images in general here and there and the skin. But tuberculosis is normally transmitted by contact by coughing. So again, this is also peculiar. For primary complex, because it's also possibly children, nobody diagnoses primary complex or did diagnose primary complex because the child was coughing. It was in a sense lymphadenitis of the lung rather than affecting the lungs by themselves, but more lymphadenitis of the lung. And the adult pulmonary tuberculosis is very, very commonly diagnosed because the persons have coughing, sputum shows presence of mycobacteria, there is weight loss, and there are other things as well. So this is something that is transmitted by the patient to the contacts around it, and this is where the modeling is important, because you know that there are contacts in the family. If the person has tuberculous organisms in the cough, they can be easily taken up. Again, look at the immune response. There have been cases, and people who have worked on tuberculosis families would easily be able easily tell you that there might be one case, there might be 10 people who live in the same family, and maybe one of them catches it. And if you really look at the exposure, it's probably similar. So what happens? How do you distinguish that this particular individual has gotten it because of X, Y, and Z factors? There are obviously many factors which will contribute to the transmission, further immune response, and so on and so forth. So when you are looking at, Sebastian asked us to one person getting up and two getting infected and 4 getting infected and spreading of the disease, tuberculosis is not easy to follow or understand. Is it from one person, and in this room if I am coughing and spreading tuberculosis, will all of you get it? Obviously not. Who will get it? I have no idea. And some people do need to figure out that if everybody here is not, healthy in the sense of not immunocompromised, I don't know who will get it, but somebody will get it. So what host factors and what other factors contribute to catching the disease is something that we really, really do not know. And if it is other kinds of tuberculosis, as in lymphadenitis or TB meningitis and others, the spread is not via aerial route, not via coughing or not via the route I described. So how do I get TB bacilli by aerosol, by aerial route, by somebody coughing onto me sort of, and how does it go to the lymph node, and why does it settle there, or why do I get TB meningitis? Where does this bacterium go and how does it go? What are the trajectories that the bacterium undertakes once it is probably in the upper lung area? No idea. I haven't come across, maybe Gwen will talk about it in terms of modeling when she talks to you a couple of days later, that what is it that people think that this is how probably transmission happens, and these are the factors that we need to take into account. I am completely blank about it. So as I said, these are just the spectrum, and let me just mention about the BCG vaccine. So far since morning we haven't talked about the vaccine, but vaccines in general, but BCG is one of those live attenuated vaccine. And how many people know what is a live attenuated? Okay, how many do not know? Easier. Whether I need to explain it or not is what I'm trying to guess. Okay, I do need to explain. So the idea of live attenuated is that the vaccination should mimic natural infection. If it is a virus or it is a bacterium, it has to be live for infection to be caused. So live goes without saying that if there is a mimic that we need a live vaccine. However, when we say attenuated, if we look at the historical times, we did not know what was different between the attenuated strain and the parent strain. From the parent strain, in some condition or the other, the virus or the bacterium was grown. We didn't have this kind of technology, really sophisticated technology that we have today about sequencing, finding out which parts are deleted, which parts are mutated, and so on. So the scientists arrived at a certain strain or a certain cultivar and decided that maybe this is attenuated enough and we can use it as a vaccine. So when we call it attenuated, it was growing pretty well, obviously, but the criterion should be that it should not cause disease. And here, the separation that I made in the morning would be useful. It should not cause disease, but it should cause infection. Make sense? Yeah? So we never ever want disease with a vaccine. So infection, for one reason or another, will stop by itself in natural conditions. I'm not talking just now about vaccine, I'll draw a parallel in a minute. But in natural scenario, infection will establish itself and will end by generating immune response. In some cases, because of one factor or another, maybe 25, infection doesn't remain self-contained, but it becomes a disease. Proper illness, morbidity, mortality, various things will happen. Now, the vaccine, live attenuated vaccine, is mimicking infection, but it should never ever reach the stage that it will go towards disease. In some individuals which are, say, immunocompromised, even BCG kind of vaccine or some other vaccines like polio vaccine, oral polio vaccine, can cause disease, or in cancer patients because they are on immunosuppressive treatment, one kind of one or another. But that is an abnormal situation in the sense that's not what the normal relatively healthy population is. So in a healthy population, live attenuated vaccine should cause infection and stop. First criteria. It should not also, rather it should produce immune response which is very similar in nature to the original infecting organism, pathogenic organism. So BCG in this case should actually produce an immune response which is very similar to what M. tuberculosis does. So remember the example of circumsporozoite vaccine, circumsporozoite protein. If BCG, for example, is producing immune response against A, B, C, D as the proteins, and M. tuberculosis is actually producing immune response against P, Q, R, S and T, then BCG is not useful, because amongst the P, Q, R, S, T is the protective response mounted. In A, B, C, D, E, there may not be the case. I'm taking this as sort of, broad generalization for understanding, but we need a vaccine which is live, which is attenuated, and it should also trigger an immune response which is protective in nature. Just having a live attenuated vaccine does not make sense. Okay? If this is the case, now one can think how good or bad the BCG vaccine is. But it's a long one. Can I stop and we have coffee? Because my throat is talking to me now. We'll come back and go with. It's not there? Sorry.